So good morning, and on behalf of Professor Kulkarni and the Jawaharlal Nehru Center for Advanced Scientific Research, may I welcome you all to this seventh Darwin lecture. The Darwin lectures were initiated in 2009 during the 150th anniversary of the publication of The Origin of Species. The first lecture was given in 2009 itself by Professor Larry Muller from the University of California at Irvine, and subsequent lectures were delivered by Professors Russell Foster, Brian Charlesworth, Jerry Coyne, Nicholas Barton, and Joachim Krug. It's a very great personal and professional pleasure for me to introduce to you today's very distinguished speaker, Professor Vassiliki Betty Smocovitis from the Departments of Biology and History at the University of Florida at Gainesville. She is a very distinguished historian of science, and in particular, a historian of the mid-20th century evolutionary synthesis and the subsequent period. Professor Smocovitis has received numerous honors and recognitions both for her teaching and her research. And I'll just mention a couple of them here. She's a fellow of the American Association for the Advancement of Science. She has been editor-in-chief of Studies in Botanical History 2005 to 7, associate editor of the Encyclopedia of Evolutionary Biology from Elsevier. She is co-editor-in-chief of the Journal of the History of Biology and is the president-elect of the International Society for the History, Philosophy, and Social Studies of Biology. Professor Smocovitis is a diaspora Greek who was born and grew up in Egypt. She is now a Canadian citizen and works in the USA. She got her bachelor's degree in biology from the University of Western Ontario and her PhD in ecology and evolutionary biology from Cornell in 1988, working with William Provine. And starting from her PhD days, she began to switch her interests from evolutionary biology per se to more about the history of evolutionary biology. In addition to being a great scholar of the history of botanical sciences in the US and elsewhere in the 20th century, she is a particularly well-known scholar of the history of the evolutionary synthesis from the mid-20th century and the period immediately afterwards that saw the crystallization of evolution not just as an overarching perspective on biology, but also as a well-defined academic discipline. Her 1996 book, Unifying Biology, The Evolutionary Synthesis and Evolutionary Biology, remains greatly appreciated and referenced. And this book also embodied many of her unique insights into the historiography of science, treating science as a discourse and a culture of its own, with its very own canonical works, rituals, narratives, and discourses. She will be talking to us today about G. Ledyard Stebbins and the path towards an integrative biology in the period after the so-called evolutionary synthesis. Professor Smocovitis, over to you.
Oh, thank you. Thank you so much. And thank you for your hospitality. This may be the last time that I get to see some of you. So thank you all. I'm sorry I couldn't make Korg, and sorry I can't be, I am live. I'm by remote and I'm not that far away. And I'm happy to say that I've been negative for 3 days. So I think I'm over this terrible, this COVID infection. So thank you very much for the introduction and hosting me and just all of it. So what I wanted to do today has 2 parts. And by the way, just again, a little bit of it is repeating what I did with Korg. I'm a historian, and historians do not have graphs, data. We do not give talks in the same kind of way. The humanities has a very different culture. So what I will do is show you slides with a great deal of images and quotations, because I cannot possibly remember those off by heart. And it's a little bit unusual, but please bear with me. Parts of it should be a little bit fun. So as I say, there's two parts to my talk today, and let me share the screen. The molecular wars and the origins of organismic biology, or how a naturalist makes peace with, quote, the Caligula of biology, end quote. So let's start with the evolutionary synthesis, just very briefly. It saw the fusion of Mendelian genetics with Darwinian selection theory in a populational view that explained the origins of biological diversity. It saw the reconciliation between different points of view bearing on evolution, and it brought together systematists, paleontologists, botanists, and geneticists, and led to a new discipline that became known as evolutionary biology. It was marked by the founding of the Society for the Study of Evolution, which sponsored the first international journal of evolution called Evolution, and a series of celebratory meetings held at places like Princeton in 1947 that brought together an international community of workers who began to call themselves evolutionary biologists. By the end of the 1950s, during the period of the Darwin centennial, not only was there a discipline that had emerged, but it argued for the unifying power of evolution within the disparate and heterogeneous biological sciences. The idea began to gain currency that evolution was the central organizing principle of biology and the unifying theory of biology. But while this struggle to unify the biological sciences appeared to be over, the struggle to preserve the unity, the autonomy, and the location of evolution and the biological sciences continued. The architects, and this is a term that is self-designated, mostly Ernst Mayr, calls himself and all of his colleagues the architects of the evolutionary synthesis, began to function as the unifiers of the biological sciences. And this is a photograph that I pull out of Futuma's 1986 textbook. And here we have the mathematicians, R.A. Fisher, J.B.S. Haldane, Sewell right at the top, Julian Huxley and Ernst Mayr at the bottom. And then we have G. Ledyard Stebbins, G.G. Simpson, and Theodosius Dobzhansky in this particular image. So what they began to do was to begin to think and to draw disciplinary boundaries. And they would say, for example, this is evolution, this is not evolutionary biology. This is close, this is far. So what they were doing was kind of negotiating the boundaries, demarcating the boundaries in a series of exchanges that they had all through especially the 1960s, as we will see. What they were doing was trying to preserve the delicate balance between the unity of biology and the autonomy. And what do I mean by autonomy? Well, biology had to be enough like physics and chemistry. It had to have the rigor, and it obviously had to logically connect to physics and chemistry, but it could not be reduced to physics and chemistry because then it ceased to be biology. So when Ernst Mayr and others begin to use the term autonomy, what they're doing is saying biology is an autonomous science. It connects to physics, chemistry. It connects to the anthropological and the social sciences, but it's also an autonomous discipline of its own. Now, everything was hunky-dory until 1953, because two events led to a destabilization of the biological sciences. In the first place, the classic, very well-known experiment by 1953, the Miller-Urey experiment for the origins of life, confirmed the biochemical basis for the origins of life. And as important, if not more so, 1953, Watson, Crick, but also Rosalind Franklin and Morris Wilkin, their work led to the articulation of the structure of the macromolecular secret of life, DNA. And I like this picture because it was part of the 50th anniversary, and you can see who is featured most prominently, and that is Rosalind Franklin in the front. As research in what came to be called molecular biology. Molecular biology was a term that was coined and introduced in the US by Warren Weaver in approximately the late 1930s. And as this area just began to intensify, and it experienced in the 1940s, biochemistry began to boom. And then the articulation of the structure aided molecular biology even more so. The linkages between the physical sciences, the linkages between physicists, chemists, and biologists solidified yet further. In fact, many of the molecular biologists, many of the pioneers of molecular biology had no training in the biological sciences. Their primary training was actually in the physical sciences. So with the subsequent articulation and the refinement of the molecular basis for genetic change, many biologists themselves began to jump on what eventually came to be called the DNA bandwagon. And so popular did molecular biology and the DNA bandwagon become that biology soon began to face a great threat to unity and to autonomy. It was threatened with complete engulfment by the physical sciences. The beginnings of the rift, this rupture that I'm going to describe in part one, between what came to be called organismic biology and molecular biology was felt by the late 1950s at institutions like Harvard University, where at least two of the architects of the evolutionary synthesis were safely ensconced. I mean, they were very well off and privileged. And this was Ernst Mayr and G.G. Simpson. In a very famous exchange with the biochemist George Wald, and he eventually got a Nobel Prize, a very, very well-known biochemist, Ernst Mayr defended the diversity of the biological sciences and the legitimacy and unifying power of evolutionary biology. And here's Ernst Mayr, one of a picture I took in 1996. In 1959, on the occasion of what has been called the Where Are We Beanbag Lecture, Ernst Mayr made an impassioned plea on behalf of evolutionary biology. And he wrote, "We live in an age that places great value on molecular biology. Let me emphasize the equal importance to evolutionary biology. The very survival of man on this globe may depend on a correct understanding of the evolutionary forces and their application to man. The meaning of race, of the impact of mutation, whether spontaneous or radiation-induced, of hybridization, of competition, all these evolutionary phenomena are of the utmost importance for the human species. Fortunately, the large number of biologists who continue to cultivate the evolutionary vineyard is an indication of how many biologists realize this. We must acquire an understanding of the operation of the various factors of evolution, not only for the sake of understanding our universe, but indeed very directly for the sake of the future of man." And let me just tell you, by 1959, Ernst Mayr was literally bristling. He was very angry, not just at the beanbag geneticists, but at people like George Wald and the fact that molecular biology was beginning to get the lion's share of grant money. There was no way that systematists in particular could compete with the kind of money that was being poured into and the kind of institutionalization that molecular biology was seeing at the time. So he felt a very real threat. Okay, nor was Mayr the only one. In his spirited defense of evolutionary biology. By the early 1960s, he was joined by these fellow unifiers, the great mammologist and paleontologist George Gaylord Simpson, who wrote eloquently, and he had been an English major. He just wrote beautifully. Eloquently, he wrote about this ensuing crisis in the biological science. And Theodosius Dobzhansky, the Ukrainian-born evolutionary geneticist and Russian emigrant to the United States. And he felt that molecular biology was becoming, quote, a glamour field, end quote. It attracted old and young alike, and they had been swayed by the belief that the only worthwhile biology was molecular biology. With time, and perhaps in response to what they saw as an arrogant, steamrolling tendency of some of the molecular biologists, this, what I call the spirited defense, began to lead to attacks, really nasty attacks in print. Especially damning was Simpson's attack on the new molecular biology in his 1964, referring to this gaudy bandwagon that traveled with this banner with a strange device called DNA. Though they may have hated some of the new biology, the architects also had enough sense to know that they had to accommodate to the new science, which had such promising explanatory power. So here we are, Ernst Mayr in his prime again. 1961, he writes the first of his philosophical papers titled Cause and Effect in Biology. In it, he made a striking revelation, comparing causality in biology with physics. He argued that causality in biology was, quote, a far cry from causality in classical mechanics, end quote. The structure of biological science was far more complex and had its own emergent properties, unlike any of the physical sciences. Biology, from then on, would consist of two biologies, functional biology and evolutionary biology. The biology of proximate causes, the biology of ultimate causes. The danger of complete reduction to molecular biology and ultimately physics was avoided and indeed made to work for biology through a unifying argument launched and articulated by Theodosius Dobzhansky. He too divided biology into two halves, which he called the Cartesian and the Darwinian halves, arguing for the integration between the two. And what he did was to incorporate the molecule as the new level of evolution. This new level served integrative functions, accounting for both the unity of life with its connection to the physical sciences and the diversity of life with its connection to organismic biology. So, physics, chemistry, and molecular biology accounted for the unity of life, whereas organismic biology, ecology, and the social sciences, on the other hand, would account for the diversity of life. Evolutionary mechanisms were responsible for both the unity and the diversity of life so that the unity of sciences was preserved. But it was the organismic biological sciences, situated between the molecular and the physical sciences and the social sciences, that would begin to answer the big question: what is man? End quote. Extending the unifying properties of evolution, Dobzhansky forcefully argued for the importance of evolutionary study to all biology, in so doing, first ushering the phrase that gained currency with evolutionists in the wake of the modern synthesis. And here's the first appearance of this famous quotation: I venture another and perhaps equally reckless generalization. Nothing makes sense in biology except in the light of evolution. If the living world has not arisen from common ancestors by means of an evolutionary process, then the fundamental unity of living things is a hoax and their diversity a joke, end quote. Well, so much for these eloquent unifying arguments. They didn't help the situation at places like Harvard at all. Graphically described by antisystematist and sociobiologist E.O. Wilson in this autobiographical book. I recommend reading this to everyone. In a chapter that he titled, appropriately enough, The Molecular Wars, Harvard biology rocked, the whole department rocked, as appointment after appointment fell to molecular biology. And it was something like 7 out of 8 appointments, new hires, were all in molecular biology. Working with the likes of this man, James Dewey Watson, AKA Honest Jim, was distasteful to Wilson, who felt both, quote, stifled and illegitimate, end quote, by the standards of the new biology. Wilson recounted that Watson was, quote, the most unpleasant person that he had ever met, and then described him as the Caligula of biology. The faculty meetings at Harvard were so, quote, edgy that Harvard biologists gathered, quote, like Bedouin chieftains around a disputed water well. I mean, the metaphors, the language is just so powerful in depicting the dysfunction and the tumult and the conflict that took place between molecular biology and the systematists and everyone else who was located at Harvard. The rising tension was due not just to the clash of, quote, megaphonistic egos, but the fissure ran deeper into the very definition of biology. Wilson wrote, the molecularists were confident that the future belonged to them. If evolutionary biology was to survive at all, it would have to be changed into something very different. They or their students would do it, working upward from the molecule through the cell to the organism. But the message was clear. Let the stamp collectors return to the museum. By the mid-1960s, the situation at Harvard was so dire that Harvard biologists took an unprecedented step. They drew up a separation agreement. Separation agreement is another word for divorce. Now that's what they did. They agreed to separate, to divorce. So working with the assistance of the Dean of the Faculty of Arts and Sciences and the chair, geneticist Paul Levine, Harvard biologists split themselves into 2 separate units. So after many conversations pertaining to the naming, would they call themselves classical biologists? Would they call themselves macrobiologists? They chose the name organismic biology. So that's how the name organismic biology emerged and then stuck. And so were born the 2 celebrated divisions of biology, molecular biology, and organismic biology. Part 2, enter Stebbins, from naturalist to developmental biologist. As unifiers, the architects of the synthesis seem to be especially drawn to the molecular wars. All, that is, except for one, this man, G. Ledyard Stebbins. Where exactly was Stebbins in these celebrated controversies? The answer? He was doing it. By the mid-1950s, Stebbins had entered the areas of developmental genetics that began to expose him to some of the biochemical, physiological, and molecular approaches that were threatening his fellow architects. Not only was he following the lead of these reductionistic scientists, he was enthusiastically embracing and celebrating these approaches. Stebbins was not just writing, he was driving that gaudiest bandwagon described by Simpson. Why exactly was Stebbins so keen on DNA? Where was he located in this disciplinary dynamic? For starts, unlike his colleagues at Harvard, like Meyer and Simpson, who were appropriately located at the Museum of Comparative Zoology, or Dobzhansky, the star of evolutionary genetics, who was in a kind of splendid isolation at the Rockefeller, Stebbins was the chair of one of the more active departments of genetics in the United States. He had to be current in the new reductionistic sciences. Hired in 1950 from the genetics department at the University of California, Berkeley, Stebbins was appointed as the first chair of the first department of genetics in the growing university campus at the University of California, Davis. And I'm not sure you're aware, you know enough about the dynamic between Berkeley and Davis. Immediately after the war, the order was given that Davis would become the primary agricultural school, so that it literally took over a lot of the work that had previously been done at Berkeley. Stebbins' charge was to build and guide the new genetics department in one of the fastest growing and what became one of the largest agricultural schools in the whole United States. He held this position as chair during this critical period, 1950 to 1963, that saw the rise in popularity of these reductionistic biologies. So during this interval of time, Stebbins helped make appointments in the genetics department, reflecting some of these exciting areas in molecular genetics. He followed these trends very closely. He read voraciously, and unlike his counterparts, he felt no threat from molecular biology. And very likely, the fact that he had spent some 15 years in the Berkeley genetics department also helped give him a vastly different perspective from the museum workers, especially the ones out at Harvard. In the 1940s, Berkeley genetics was leading the way in areas like biochemistry, physiology, and virology. It hosted some of the great pioneers in molecular biology, people like Joshua Lederberg, and Beadle and Tatum's work on Neurospora crassa received a great deal of attention at the UC Berkeley campus. A figure no other than Richard Goldschmidt, himself the author of Physiological Genetics, eventually joined UC Berkeley. And Goldschmidt and Stebbins were in fact co-organizers. They got along magnificently of this group of people called the Bay Area Systematists. And I can't point, but Stebbins is in the front with the white khaki pants and the dark shirt, and Goldschmidt is in the back towards it's my right, wearing the bow tie and bald hair and having the little handkerchief, extremely well dressed considering this was Placerville Forest Station. So this was actually a field site. So you can see, I mean, they're getting along very well, whereas Ernst Mayr and Goldschmidt kept fighting all through the 1940s and the 1950s. So while Meyer and Simpson had very little contact with these areas of research, Stebbins was in the thick of things, even as early as 1940s in a department renowned for its forward-looking genetics research program. Unlike them too, Stebbins had a lifelong tendency to follow new and exciting currents of scientific research. No one studying his career could accuse him of being old-fashioned. Indeed, his career followed the currents unfolding in general biology, usually as they applied to the plant world. And although he was trained in floristic botany, and this is, looking at the distributions of plants, classical herbarium systematics, he could do that kind of work. And he began actually his career at Harvard in the celebrated Harvard Gray Herbarium. He rapidly moved to newer and more promising areas of cytogenetics while still a graduate student. And after graduation, he continued to study cytogenetics, especially as it applied to elucidating complex phylogenies in complex genera like Crepus, the hawkbeard plant, distinguishing himself as an expert in polyploidy, and in particular how it, alongside apomixis, a form of asexual reproduction in some plants, and hybridizations could serve as adaptive functions in plant evolution. So after moving to the University of California, Berkeley, where he was first appointed as junior geneticist to E.B. Babcock, Stebbins began to take the lead in evolutionary studies. And although he preserved this interest in systematic, actually for his whole career, he followed the work that was coming from breeders and geneticists, people like I. Michael Lerner, and of course, Theodosius Dobzhansky. And Dobzhansky and Lerner were close friends because they were both Soviet refugees. And Dobzhansky used to travel to UC Berkeley to visit Michael Lerner, usually speaking in Russian. But Stebbins learned a lot of the newer genetics and evolutionary genetics, listening to the two of them talking to each other. So by the mid-1940s, his voracious reading and the ability to just draw disparate literature together helped him to emerge as a leader in evolutionary studies. And unlike nearly all of his colleagues in botany or plant evolution, Stebbins knew equally well the literature on the animal side, so he could help build the bridges between the two. Very few people could speak insect evolution with Dobzhansky as an example. And Dobzhansky himself was no slouch. He also read much of the plant evolution literature. So when the two of them came together, what you had was a real meeting of the minds. And in another paper, I've argued that it's a kind of a co-evolutionary relationship that formed between the two of them. It was Dobzhansky who introduced him to L.C. Dunn, who invited him to give the prestigious Jessup lectures at Columbia University. And the Stebbins lectures for 1947, which were actually a compilation of lectures from a course that he had been teaching in evolution on the Berkeley campus. Those lectures he put together, and he published the book, Variation and Evolution in Plants, in 1950, and that book is considered the last publication in what comes to be called this critical active period within the historical event called the evolutionary synthesis. So his contributions to plant evolutionary biology are quite well known, but less well known is his research program that he began shortly after writing the 1950 book that took him to areas where he had to learn new molecular techniques. That's when he began to work with a small group of graduate students, and he began to take a direction into what he thought was the next great exciting area of both genetical and evolutionary study, namely gene action on morphogenesis. And this was the work that began to set him apart from the other architects of the evolutionary synthesis. As early as 1959, Stebbins began to map the next exciting areas for botanical study. To Stebbins, the last 15 years of biochemical genetics challenged some of the older Mendelian genetics. For example, he challenged the idea that the origin of a new character emerged by mutation or the emergence of a new type of gene. That he felt was too inadequate. He believed instead that new types could emerge not just by mutations, but by processes such as macro recombination between gene systems, especially in interspecific hybrids, which formed readily in plants. Such changes could lead to basic character changes, such as the number of parts of the flower which were, I mean, this is an important criteria or characteristic in the plant world. Stebbins thought that many of these structural differences on which students of plant evolution place such great emphasis, for example, nodal anatomy, the structure of the inflorescence, the number of sepals, petals, stamens, carpels per flower, the degree of union of some of these parts, and the nature of floral symmetry, all these were determined by an intricate pattern of balance between gene-controlled cellular sites. These, he thought, could either promote either intense mitotic division of undifferentiated meristematic cells, or they could lead to the slowing down or cessation of cell division accompanied by cell enlargement and differentiations. Stebbins conjectured that genes probably controlled these processes in at least in part through the medium of growth controlling substances, plant hormones, which were incidentally only discovered in the mid 1940s. This is all fairly new work in plant physiology. So differences in developmental pattern could be produced by altering the concentration of these growth substances, the time of release, and the rate at which they diffuse through the plant body. For Stebbins, who followed the work of plant physiologists like Wardlaw, Melville, and others, it was apparent that consideration of developmental physiology tied to genetics had the potential to shed light not just on simple morphogenesis, but then logically to evolution and thence to taxonomic considerations. So again, the systematics was always there. Small quantitative differences in the concentration, the timing of production, or the rate of diffusion of growth substances, all of which could be determined through gene action, could alter the developmental pattern. This in turn could produce profound qualitative changes in the adult characteristics of organisms. The fact that Stebbins was a botanist is critical for understanding this shift to morphogenesis. For Stebbins, plants were practically ideal study organisms for getting at some of the key unsolved problems in evolutionary genetics, much more so than animal systems. For one thing, many were easily grown, both as whole plants and as organ or cell cultures. The overall architecture of the flowering plants, furthermore, was sufficiently similar that significant observations made upon one species would likely be valid for all, with minor modifications. So too, the various parts of the plant were reproduced serially and repeatedly in a single individual, so that the effects of a single gene difference or a single alteration of the environment could be studied quantitatively on the same genetic material. Finally, development in higher plants was less complex than in higher animals, and in most instances, cellular and nuclear divisions at critical states could be more easily observed. This latter aspect was of the greatest significance in understanding gene action in generating morphological differences. While animals had but one embryonic state at the beginning of life, plants preserved meristematic tissues throughout the life of the individual. This made them virtually immortal. Plants were constantly giving a rise to new organs, of various types. Consequently, while in animals, the action of a particular gene in an individual was confined to a particular time in development, as well as to a particular change, the action of genes in plants was not time-specific and also could be stage-specific. It could take place many times in the life history of the individual, making it easily subject to experimental procedures. So, beginning in the late 1950s, Stebbins began to turn seriously to problems of developmental morphology. And by the mid-1960s, he was engaged in a far more sophisticated study of the dominant hooded gene in barley. And this is the photograph is picturing him with barley, Hordeum vulgare. So it was a good study organism since it bore 7 pairs of large chromosomes and the linkage maps had been worked out heavily. He went to work with other students on a number of other studies through the decade of the 60s, showing that mutations of genes coding for structural proteins did in fact have profound effects on morphogenesis. How did these studies play out in evolution in the context of the molecular wars? Let's go back to that. Compared to the other architects, they almost bordered on the heretical. In an essay of 1968 titled Integration of Development and Evolutionary Progress, Stebbins laid out his project for integrating evolution and development. And here he says, the most important feature of the modern synthetic theory of evolution is its foundation upon a great variety of biological disciplines, but the contributions were unequal. Certain disciplines, such as population genetics and paleontology, by their very nature provide a disproportionate share of information, but others have as yet contributed less than their appropriate and potential share of facts. One of these was developmental biology. He stated at the outset, let me just continue here. Stebbins did not have in mind the older embryology. When he thought about development, he very clearly separated it from the older embryology, which was associated with names like Haeckel and von Baer that connected ontogeny and phylogeny. And this was literally a morass, a minefield. In a well-known essay, he laid out beautifully the players and the positions played out in biology and how all of these disciplines could actually work together. Development, population genetics, evolution, all of them had their place. And let me read you this marvelous quotation, 1965. Biologists and laymen alike often try to answer the question as why does the giraffe have such a long neck? The answer which a biologist would give to the question would depend on his training and interest. A field naturalist might reply, because this fits the giraffe to his environment by enabling him to get food from the leaves of tall trees. A Darwinian evolutionist would say, because in the remote past, certain animals produced more offsprings to which they transmitted their longer necks. The answer of a developmental biologist might be because in the embryonic and fetal development, a large quantity of growth hormones concentrated in the neck region, thus stimulating the excessive development of the 7 neck vertebrae and the tissues associated with them. Finally, a modern molecular geneticist would be likely to answer because a part of the code of the DNA of the giraffe's nuclei carries information for a long neck. To Stebbins, none of them was actually wrong, and none of them was more basic or more important than the others. Each was essentially correct, but incomplete. Surprisingly enough, he wrote, however, one can find sophisticated, high-level discussions of similar biological problems in which one of the 3 types of answers is considered to be the only significant biological one, and the others are minimized or neglected. Even when the need is recognized for these different approaches to such basic problems, each type of answer is often regarded as sufficient in itself. So throughout the epoch of the molecular wars, Stebbins actually did not write a single essay attacking molecular biology, nor did he write essays with impassioned pleas on behalf of systematics or evolutionary biology. Instead, there was enthusiasm and sometimes a celebration of the new biology. In only 1964, for instance, on the occasion of his presidential address to the American Journal of Botany, he wrote, let me just here, classical and molecular biology are not basically inimical, but complementary to each other. Moreover, whatever may be the approach which any botanist prefers, he will inevitably be more effective in his own approach if he understands as fully as possible other phases of the same problem. For botany, as well as botanists, the key to real progress are diversity, integration, and mutual understanding. These are not empty words of rhetoric aimed at diffusing the molecular wars. He meant them and continued to mean them well into his last days, trying very hard to follow the currents of biology. And sadly enough, I located an enormous manuscript which attempted to integrate molecular biology, development, and evolution. He wrote it sometime in the late 1960s, titled Molecular Order and Development, and it appears to have been rejected. Nonetheless, Stebbins' attempts at integrating diverse approaches carried him far. He collaborated with a set of workers outside the traditional circles of the architects, becoming friends with people like Conrad Waddington, seen here in the photograph. Waddington has the pipe and is the bald man in the middle of the photograph. And here they are at the International Union of Biological Sciences in Vienna. They also worked together on the International Biological Program. Throughout the 1960s, Stebbins reflected the emergence of an integrative biology capable of engaging biologists from a range of disciplines. He continued this integrative approach in his research efforts too, but also in his popular essays and textbooks, very influential textbooks like Processes of Organic Evolution, and in helping to establish national and international textbooks and teaching initiatives like in biology, like the biological sciences and curriculum study. His last book reflected his lifelong ambition to unify and integrate biology. It was titled Darwin to DNA, Molecules to Humanity. Okay, closing thoughts. What general conclusions can we draw from examining Stebbins in the context of the molecular wars? And why did he appear to be so removed from most of the controversies? For one thing, he was located as chair of this dynamic genetics department that was expanding rapidly. And for another thing, he displayed a lifelong career tendency to shift to exciting new areas of research. I'm not surprised, having studied his life, that he moved to developmental genetics in the 1960s. That was typical for him. As a botanist as well, he worked on an organismic system that held special promise for elucidating the gene to character transformation. And perhaps, too, it was part of his character that he never engaged heavily in these public controversies. He had a terrible temper tantrum and argued with people, but it was never in one of these public settings. Contrary to historical perceptions, furthermore, there was no huge rift between evolutionary biology, the architects as a whole, and developmental biology. Too much has been made, especially recently, by advocates of the extended evolutionary synthesis, that there was this absence of development, that it was excluded from the evolutionary synthesis. And I would argue that in part, they think this because the emphasis they place is usually on animals, not necessarily on plants. And they don't look at Stebbins. They look at everyone else who was controversial and arguing and defensive and territorial. His own career attempted to bring development and biology together took place immediately after he finished the book that published in 1950. So, it's not as though developmental biology was actively excluded from the synthesis. It was the next great integrative step. Indeed, the word integration began to substitute for the word synthesis in much of Stebbins's own work, as it did for many of his colleagues at the University of California. In the mid-1960s, for example, Stebbins was part of an integrative biology studies group that the first formal department of integrative biology, which took a number of these life sciences units together on the Berkeley campus in 1993, that you had this first department of integrative biology formed there, is not surprising. Integrative perspectives often meant the application of molecular techniques to solving long-standing problems. And here is a photograph for you, food for thought, because this was the iconic image chosen to represent the whole of the University of California that had its 100th anniversary. And it's a photograph of Stebbins with women and people of color. And I don't think it's an accident that the term integration gains currency in the biological sciences in the 1960s, because the word integration was on the tongues of many of the US civil rights leaders. So it's integrative biology, very little of the synthesis. They're not engaged in a synthetic project. They are engaged in an integrative project. So let me begin to really wrap it up and say, as historians of science, those of us who study the history of science, we actually have a lot to learn from Stebbins and the molecular wars of the 1960s too. And for a long time, the history of science told us of heroic individuals leading on the road that led inexorably to truth, to something called truth. And historians and scientists wrote the simple history of the victors, erasing the losers, the twists and the turns, and the debates from which a negotiated perspective emerged. And this older historiography was replaced by a view that science takes place in collectives that frequently clash over competing interests. A view best demonstrated, for example, by historian Martin Rudwick's The Great Devonian Controversy, philosopher David Hall's book, Science as a Process, or sociologist Bruno Latour in books like The Pasteurization of France or Science in Action. The molecular wars appears to have been a classic case demonstrating that the kind of collective wars and territorial feuds we see in Rudwick, Hall, Latour, and many of these feuds that others have described is, there's diversity in the ways that science is engaged and that science is practiced. But the truth is that much of the history of biology has left out Stebbins and his research program for this last part of his career. And I think an examination of his part or his non-part, he wasn't a participant in these molecular wars, may teach us that sometimes concentrating on sensationalism, on controversy, on arguments and fight may not be in the best interest of history. So I would close with saying that with this kind of historical hindsight and with currents that call for a continued integration of evolution, ecology, and development, what is called Evo, Eco, Devo, using techniques from molecular biology, Stebbins may very well have been right way back in the 1950s. So thank you very much. Thanks for your patience.
Thank you very much, Professor Smocovitis, for a wonderful, wonderful talk. Sorry, were you saying something?
Well, I was just saying I took this photograph of him, and I always like to show it because it's one that I took. And this is where this is part of his personality. in this lecture at Cornell, what he does is to say, I was wrong about the species concept in 1950, which is one of his many charms. And he says, you know, he was just completely wrong. So can we get out now? Okay.
Yeah.
Questions? But you can
Yes,
see, just to pick up on our the 7th Foundations of Biology, the theme of the extended synthesis. Now you can understand why I get frustrated, because they just don't know enough about Stebbins and what he was doing.
Yes, indeed. Maybe you could stop sharing the screen so that we
Oh, I thought I had.
we see
Okay.
you better.
Okay.
And
That
wonderful.
better?
So we do have time for some questions. If people want to either raise their hands or maybe write questions in the chat box, I can read them out.
Satya Bratha?
Yes, Satya, go ahead. Unmute.
Am I audible now?
Yes.
Yeah.
Please go ahead.
So the question is in somewhat of a parallel to what was discussed today. The way you described this territorial conflict between the molecularists and the classical evolutionary biologists at the inception of the molecular sciences. Is there a parallel to another so-called encroachment of explanatory encroachment of physical sciences onto biology, which is biophysics or biophysics today? They don't necessarily take the genes path to explanatory encroachment, but they do provide an alternative or a complementary explanatory method for certain biological phenomena. Is there a conflict going on today or at the inception of that?
Oh, no. In fact, the irony is that museum workers are genomicists. All the PCRs are running in systematics. It has quite literally given them, it's a game changer. And so a lot of this tension now has been forgotten. But what I wanted to come back to you, I mean, I have studied, there is another famous dispute with physical sciences, and that is over exobiology. Exobiology was the science, there's a term that was coined by Joshua Lederberg to denote the biology of extraterrestrial organisms. And this is in the context of SETI, the Search for Extraterrestrial Intelligence. And there's all these conferences that are held by organic chemists, by people like Carl Sagan, astronomers, and Frank Drake. And they think they're studying evolution. In fact, something you may not know, Carl Sagan's first article appeared in the journal Evolution. He was a student of H.J. Muller's. So Meyer and Simpson entered literally at the same time. This is in the early 1960s. And just, it was fireworks. And one of the reasons being, again, you don't know anything about biology. You're a bunch of physicists, you're a bunch of chemists. You don't understand the process of evolution. There's fights between Lynn Margulis and many of the microbiologists and people like Ernst Mayr, because they're looking at different levels of evolution. And, when she comes up with her endosymbiotic theory, he literally throws a fit. So all through the 1960s, there are many such instances of where they're ganging up. Do you know what I mean by ganging up on physics and physicists and chemists? And there's this culture where they look at somebody like Schrodinger's 1944 book, What is Life? And they laugh because Schrodinger knew nothing about biology. So there's real tension. And some of this has to do with the fact that, of course, physics after the Second World War has gained they're the architects of the atomic bomb. They control the planet, basically. They are a very high status field. The resources go to them, the attention goes to them, and these poor biologists have a very hard time establishing themselves. And just one more thing, does it exist today? I was a postdoc at Stanford in I think it was 1992, the year, and I was having breakfast with David Botstein, who was in charge of the Human Genome Project. There was a small meeting, and I was invited because, again, you know, I'm a historian. They want me to observe this history. And I remember one of them came in, I think it may have been, it was a woman who said, well, the National Science Foundation just killed the superconducting super collider. And this bazillion-dollar project that was supposed to go to the superconducting super collider was killed. They weren't going to be able to build it. And the Human Genome Project was, of course, this was 1992. This was, you know, at the peak of the promise. And I remember them cheering, you know, because biology finally was getting the attention that it deserved. So I do think, I mean, there's a lot of this kind of silliness and competition. But I think, to come back to a little bit more serious answer, I do not think the tension exists now. I see many people using molecular techniques to solve, you know, long-standing problems. Does that answer your question?
Yes, absolutely. Thank you.
Any other questions? Ben, yes, please go ahead.
Hi, thank you. Yeah, that was great. My question is really as follows on from what you've just said, really. But at the time, and perhaps outside of California, what was the sort of molecular side of the molecular wars like? Were they, you know, is it fair to characterize them as primarily not being very interested in organism camp, or were they hostile themselves?
No, I don't think they were. I think they just, it was a separate worlds. I don't think molecular biologists cared very much, except unless when you're talking about people like Joshua Lederberg. Lederberg was a very serious biologist. He was interested in many different kinds of things. But if you look at, for example, Cold Spring Harbor outside of California, and by the way, this isn't a bias. You know, I've looked at degree programs after the Second World War, and what happens is California gets a lot of resources after the Second World War and begins to hire people. And there's a tension in the US between the West Coast people and the established private elite school. And you know, the University of California is a state school, and they have a very different ethos, and they collaborate. The departments are, you know, mixed with each other. They're co-teaching courses. Whereas if you go to Harvard, they are so elite. They have so much money. They all have fiefdoms. But let's go to Cold Spring Harbor, and I would say that most of the people there just didn't care, or they looked down their noses at the stamp collectors, the systematists. I picked this up in graduate school.
But then, so follow on from that, I guess, then this educational effort of Stebbins, do you think that was successful in pushing back against that?
Absolutely, absolutely. His educational efforts? Yes, I do. Yes, I do. I think the biological sciences and curriculum study, and incidentally, it was international. I don't know if it existed in India, but I know it existed in Japan, and it existed in Canada. I am a BSCS baby. I turned to evolution with a lot of people having been literally, what's the word? I'm not going to say, I'll say educated. What I think evolution is, from the origins of life, beginning with the experiments of people like Oparin in the Soviet Union, and the making of these proteinoid microspheres, and then going through, but having this idea of the biochemical basis for the origins of life, to me, this was a given. So I never experienced that kind of tension, but I'm a 1970s baby. I came the decade afterwards. But that textbook series, Ben, was actually written. Stebbins was involved in that project. And this whole argument that evolution is the central unifying, the central organizing principle, nothing in biology makes sense in the light of evolution, that dominates virtually all the textbooks of biology after Dobzhansky. And it's used, it appears again on the cover of the American Biology Teacher in 1975, because the creationists are beginning to argue against the teaching of evolution. So evolutionary biologists come in and say, you can't teach biology without evolution. It's ridiculous. So it was effective, yes. I think
Thanks,
there's a question.
Yeah, there is a question from Rutvich Kulkarni. I'll just read it out from the chat box. He's asking, do we have any historical evidence for ideological debates in science resulting in some disciplines actually delaying their development, perhaps due to rejection of valuable work that was, quote, not in line, end quote?
Oh, of course, and the difficult word here is ideology. What do you mean by ideology? And here, let me further kind of open it up for discussion. I mean, the whole biometrician Mendelian debate, I'm not sure I'm comfortable calling it ideology, but that was one of the more counterproductive episodes in the history of science. And they had dug in. So yes, there are many such instances where people were arguing and engaging in fruitless, silliness and debates. Many cases.
Any other questions?
So I guess the moral of the story is you really need to keep your mind open, and this is one of the reasons that this whole EES, the extended evolutionary synthesis, makes me a little bit queasy, suspicious, because it's a fight and anytime I see these fights in the public, in the press, and strong personalities, and what Wilson calls megaphonistic egos. Anytime I see that, I begin to wonder, is this another example of a fruitless debate?
Yes, absolutely. I couldn't agree more. Let's see. If there are no further questions, then let's thank Professor Smocovitis once again for a wonderful talk and for some interesting insights afterwards in the chat session. Thank you very much.
Well, thank you.
And
you can tell I enjoy when I read these things, and I have abbreviated this. It's actually very long. If you read the things they say to each other, the insults that they heap, creative
yes.
insults. You can't
Yes.
believe that these are great scientists.
Yes. So wonderful. We will close the meeting then, and thank you so much once again.
Well, thank you. Thanks for coming.
Yeah.